this topic, we're going to have a look at natural selection. So we're going to discuss what is natural selection and, of course, look at Darwin. Then we're going to have a look at a few case studies: medium ground finch, antibiotic resistant bacteria, sickle cell anemia, pesticide resistance, and then finally industrial melanism. So what is natural selection? This is the process by which organisms that are better adapted to their environment survive and breed, whilst those less well adapted fail to do so. So here you can see mice that are not camouflaged will be eaten, whilst the camouflaged mice will be able to breed. So predation by the bird of prey is what we call a selection pressure. And selection pressures increase the changes of some alleles being passed on to the next generation and decrease the chances of others. So in this case, the alleles for dark fur have got a selective advantage over the alleles for white fur. Now in the 1800s, Darwin visited the Galapagos Islands. These are volcanic islands about 5 million years old, and they've got many unique species on them. He collected 13 different types of finch, which are birds, and he noticed that these birds had different beaks due to what they ate. So if you have a look here, you can see that these finches are closely related, but they differ in beak sizes and shapes. So Darwin found that each beak is adapted to what the finch eats. This helped him to formulate his theory of evolution. So his theory of evolution is that many species of finches were derived from a single species that had adapted over time to the different environments. This is called adaptive radiation and divergent evolution. So have a look at what these finches eat and how their beaks have been shaped. So through his studies, Darwin developed the theory of evolution based on a few principles. All organisms produce more offspring than can be supported by the supply of food or light. Despite the overproduction of the offspring, the populations remain constant in size. This means that there must be some competition between the members of species so that some can survive. So if you look at this example, we've got different beetles, orange and green beetles. They're going to compete for food, light, space, but somehow their numbers remain constant. So you can see that in this population of beetles, there are two types, green and orange. This means that they are slightly genetically different. You can see that the birds prefer the green beetles. This means that the allele for the orange color makes the beetle survive better. So as time goes by over many generations, these orange beetles will be able to survive and breed so that they can pass on the allele for orange color. Over many generations, the frequency of the favorable alleles, which is the orange color, in the population will increase. Right, let's have a look at a few case studies as examples of evolution due to selective pressure. The first is the medium ground finch, antibiotic resistant bacteria, sickle cell anemia, pesticide resistance in insects and mammals, and then finally industrial melanism. Okay, starting off with medium ground finch. Peter and Rosemary Grant visited the Galapagos Islands, in particular the island called Daphne Major, which you can see here. They studied finches on this island for many years and noted down the average beak depth. So you can see the average beak depth in 1971 of the birds. And have a look at this graph. Then in 1977, there was a drought on the islands. This means that there was limited food for the birds, and the only seeds available were large and difficult to crack. So when they returned to the island, which bird do you think predominated? The birds with big beaks that could crack these large seeds, or the birds with small beaks? As you guessed, they found that 90 of the birds that survived, more of them had bigger beaks. So if you have a look at these graphs above, you can see that the graph at the bottom has been shifted to the right. That curve has been shifted to the right. This shows that a selective pressure, such as drought, has shifted the graph to the right. So we say that the birds with bigger beaks had a selective advantage over those birds with smaller beaks. So they are more likely to get enough food and survive, to reproduce and pass on these alleles. Now, since beak size has got a genetic component, beak size is inherited. So the graphs showed that there was a change in beak size over time in the population, which is an example of evolution of a characteristic. Okay, let's have a look at antibiotic-resistant bacteria. So antibiotics are chemicals produced by living organisms which inhibit or kill bacteria, but don't normally harm human tissue. And most antibiotics are produced by fungi. So the first antibiotic discovered was penicillin. And penicillin stops cell wall formation in bacteria, so it prevents the bacterial cells from reproducing. So if someone takes penicillin to treat a bacterial infection, the bacteria that are sensitive to penicillin will die, and in most cases this will be the entire population of the disease-causing bacteria. If, however, there are bacteria that have an allele that gives it resistance to penicillin, they're going to survive. So there are some bacteria that have got an enzyme called penicillinase, which inactivates penicillin. These mutant bacteria have a selective advantage. Those bacteria without this allele will die, so they've got a selective disadvantage, whilst those that are resistant will survive and reproduce. So over time, you're going to have a new population emerging with antibiotic resistance. Antibiotic-resistant strains of bacteria are continually appearing. So alleles for antibiotic resistance often occur on that circular DNA called plasmids. So here you can see how the drug-resistant or antibiotic-resistant bacteria can pass on its plasmids to other bacteria, so that you get a different species of bacteria now becoming resistant. So have a look at this advert. Why should we not take antibiotics for colds and flu? This is because they are caused by viruses. So taking antibiotics which only work against bacteria will have no effect. And the more we use antibiotics, the greater the selection pressure we exert on bacteria to evolve and resist them. Resist these antibiotics. So here you can see an agar plate with bacteria called E. coli growing on it. If you want to see which antibiotic will kill the bacteria, you can put little discs, which have got antibiotic in them, onto the plate, that agar plate of bacteria. Where the antibiotic works, you'll see a clear area around the disc. So have a look at this plate. Which two antibiotics do not have an effect? Yes, you can see that the bacteria can grow in the presence of these two antibiotics that I've highlighted in red. This means that the bacteria are resistant to this antibiotic. Okay, right, let's have a look at sickle cell anemia. Just to recap, sickle cell anemia is caused by a point mutation in a single gene which codes for hemoglobin. So the red blood cells get pulled out of shape and form sickle-celled shapes that get stuck in small capillaries when the red blood cell is not carrying oxygen. So if you represent the normal allele as HN and the sickle cell allele as HS, now remember that the sickle cell allele is recessive. If the person has got the homozygous genotype, so it's homozygous recessive, he will have sickle cell anemia. And people with sickle cell anemia are less likely to survive and reproduce, and most die before reaching a reproductive age. Now, interestingly, if you have a look at these two maps, the one at the top represents areas where malaria is endemic, and then the one at the bottom shows you the areas where the incidence of sickle cell anemia trait is found. So in some parts of East Africa, almost 50% of babies born are carriers for the allele for sickle cell anemia. Remember that malaria is caused by a protoctist parasite Plasmodium, which can be introduced into a person's blood when the infected mosquito bites. And the parasites enter the red blood cells and multiply inside them. So people who are heterozygous, so they've got HSHN, for the sickle cell trait, also have got moderately good resistance to malaria because some of their red cells are misshapen and deflated, but they rarely develop the severe life-threatening anemia and related problems typical of homozygous sicklers. Now, those who are homozygous dominant, so they've got HNHN, produce normal red blood cells, which makes them excellent hosts for malaria. So in malarial environments, nature is going to select for which genotype. It's going to select for the heterozygous sicklers, so HSHN. At the same time, it selects against homozygous normal. So we call this a selective pressure. So just to recap, if the genotype is homozygous for sickle cell anemia, the person will be anemic and will probably not survive to reproduce. If the person is heterozygous for the allele, he will be a carrier who does not suffer from malaria and is likely to survive to reproduce. And then in malarial areas, if a person is homozygous normal, he's got more chance of getting malaria, so probably won't survive to reproduce. Okay, pesticide resistance in insects and mammals. So pesticide resistance in insects is similar to antibiotic resistance in bacteria. Insect populations have developed resistance to insecticides such as DDT, and many common insect pests are now resistant to many insecticides through their ability to break down the insecticide with enzymes, whose production has arisen as a result of a chance mutation. So in the presence of the insecticide, insects with the mutant allele are more likely to survive to breed than those with the normal allele. So the frequency of the mutant allele in the population will therefore increase. And finally, industrial melanism. Now, some species of organisms have got two or more distinct forms, which we call morphs. These different forms are genetically distinct, but exist within the same interbreeding population. So an example is the peppered moth. This is a night-flying moth which spends the day resting underneath the branches of trees. Now it relies on camouflage to protect it from insect-eating birds. It existed only in the natural light form, which you can see on the right, until the middle of the 19th century. Now around this time, a melanic or black variety arose as a result of a mutation. These mutants, the black form, had undoubtedly occurred before, but they were highly conspicuous against the light background of the tree, so they were eaten. Now, during the rest of the 19th century, the numbers of these black moths increased dramatically in industrial cities. Why? This is because the buildings and trees were blackened by soot, so these black moths could be camouflaged. So this is an example of a selection pressure which is selective predation by birds favouring individuals that lie on one extreme or the other of a range of different colour types. So these two populations of moths can interbreed, so they are still the same species. Now, changes in environmental factors only affect the likelihood of an allele surviving in a population. They do not affect the likelihood of such an allele arising by mutation. Okay, in summary, we looked at what is natural selection. It's the process by which the organisms that are better adapted to their environment survive and breed, whilst those less well adapted fail to do so. And we looked at Darwin and a study of finches. And then we also looked at different case studies, the medium ground finch, antibiotic resistant bacteria, sickle cell anemia and malaria, pesticide resistance, and then finally industrial melanism. And that concludes our lesson. The end.